Your quiz is, you see the green text, practice quiz in assignment section. So this is the assignment section you see here. So if you click there, you should be able to see that there. Okay. Anyway. Okay, so cellular level of organization is one step higher than chemical level. Chemical level is the smallest level of organization, and then one level higher is the cellular level. So first we'll talk about the cellular level of organization, and this part includes the cells, structure of the cell, and the organelles present inside the cells, and intercellular junctions. That means how the cells get connected to each other, get attached to each other. We will also talk about cell junctions. Then we'll talk about transport across the cell membrane. So how the chemicals pass through the cell membrane from one side to the other side. That's the transport across the cell membrane. Then we'll talk about tonicity. What does hypotonic, hypertonic, and isotonic mean? We'll see that. Then we'll see if you put a living cell in those three types of solution, hypotonic, hypertonic, isotonic solution, how that living cell will react or respond. We'll see that. Okay. So, how to define a cell? Cell is the smallest structural and functional living unit. So, it is the smallest living unit. That means what? An individual cell can stay alive. You know that there are many unicellular organisms, like bacteria, right? Amoeba. They can stay alive, although they are single-celled creatures. So, single cell can stay alive. But if you break that cell, then it will die. So that's the smallest unit that can stay alive individual. So that's why we say cell is the functional unit. It can function individually, right? In human body, we have 50 to 100 trillion cells, not million, not billion. Just think that trillion of cells. Over 200 different types of cells are found in our body. How they are different? They are different in size, they are different in shape, how they look like, they are different in functions. So, if you see the size, shape, and functions of cells of our body, we can divide, we can see that more than 200 different types of cells are present in our body. All cells composed of mainly CHO and N. You remember in last class when I talked about the chemical formula of proteins, carbohydrates, fats, CHON. If you see N, that means it's the whole thing is the protein. If only CHO, that means it could be carbohydrate or fat, and CHON is protein. So we know that our body mainly consists of proteins, carbohydrates, fats. So those chemicals are the main chemicals in our body. You see here, although we have more than 200 different types of cells, they just have shown few different types of cells just to give you an idea that how they can be different from each other. So let's quickly go over the topmost left one. Those cells are called fibroblasts. Fibroblasts are connective tissue cells. So fibroblasts are connective tissue cells and they produce fibers. So you see the two cells there and some fibers around them. Okay? Then next to the fibroblast, you see those two round red colored cells. Those are red blood cells. Red blood cells look red because of hemoglobin, and they are like donuts. You know donuts, you eat donuts, their shape is like a donut. And the function, what's the function of red blood cells? Transportation of oxygen. So you see the fibroblasts produce fibers, red blood cells transport oxygen, so they are different in function too, not only shape or how they look like. The third one is cuboidal epithelial cells. You know that you have already seen in last lab that cuboidal epithelial cells. And then if you go down, this is the skeletal muscle cell, very long, many nuclei inside. You already know that. So skeletal muscle cell look like this, and these are smooth muscle cells, cylindric, spindle shaped. Both ends are pointed, and middle part is white. They are much smaller than the skeletal muscle cell. Then function of muscle cell is contraction. They contract, and that causes the contraction of muscle. Then if you go down, the bottom left one, that's a fat cell. Fat cell has a nucleus, but the nucleus is not in the center but attached to the membrane. And inside the fat cell, most part is empty. Then next to that is a giant cell that is called macrophage. Macrophage is a phagocytic cell. That means they can engulf or eat microorganisms or other cells.
What's it called again?
Macrophage. You see on the top, it is there. Okay. They have what kind of property? Phagocytic property. That means they can engulf the microorganisms or dead cells of the body. Then on the right side, the top one is a neuron. Neuron is a nerve cell. And you see, neuron has a cell body that contains nucleus and a number of processes. Processes means, extensions from the cell body. And function of a neuron is transmission of electrical signal. Just write it down, that transmission of electrical signal. Then the last one, you see like a baby fish. That one is the sperm. That's how the sperm looks like. It has a head, a body or meat piece, and a tail. And the tail helps the sperm to swim in the semen. So they can really run or swim fast in the fluid. So you see that how the cells are different in shape, size, color, and functions. Okay, now we'll talk about a typical cell. If you see the structure of a typical cell, a cell has a membrane, that's the outer boundary, that is called plasma membrane. And inside the cell you will find two things. The fluid part that contains organelles, that part is called cytoplasm. So fluid part that contains different organelles. What are the organelles? Mitochondria, Golgi apparatus, endoplasmic reticulum, ribosomes. Those organelles float in the fluid, and that fluid and those organelles together called the cytoplasm. And then in the center of the nucleus, usually in the center, but not always true, you have seen in the fat cell it is not, a nucleus is located in a typical cell. So those are the parts of a typical cell. Now, nucleus is called the control center of the cell. Why nucleus is called the control center? Because nucleus contains DNA in the chromosomes. You have chromosomes inside the nucleus, and nucleus, in the chromosomes you have DNA. And DNA, you know that, control the function of the cell. That's why it is called the control center, nucleus. Okay? So this is a beautiful picture of a cell. Although if you see a cell under the microscope, nobody will, color this beautifully. When you look under the microscope, it doesn't look like it's beautiful, but it's nice to present to you to understand different parts by different colors. But the actual thing is very different. Just keep that in mind. Okay. Anyway, so this is the cell membrane. Okay, and that's the nucleus. And this part is the cytoplasm. Okay, and in the cytoplasm you have a number of organelles. I'll just mention a few organelles. Since this is anatomy and physiology class, I'm not going to talk a lot about the cell, because in intro bio you have definitely learned about the cell. So just quickly I'll go over. So in the cytoplasm you have different organelles. Important organelles are mitochondria, endoplasmic reticulum or ER, Golgi apparatus. You see those red dots? Those are ribosomes. Okay, so those are some important organelles. Mitochondria is called the powerhouse. Okay, so mitochondria is called the powerhouse of the cell. Why mitochondria is called the powerhouse? Because mitochondria produces ATP, adenosine triphosphate. So ATPs are the source of energy. So mitochondria, since mitochondria produces ATP, it is called the powerhouse of the cell. Okay. Then you see next. So if I ask you in the test which one is the powerhouse, that's the mitochondria. Why? Because ATPs are produced in the mitochondria. Metabolism occurs in the mitochondria. This is the blue tube-like structures. These are endoplasmic reticulum. And endoplasmic reticulum has two parts. Smooth endoplasmic reticulum or smooth ER, and rough endoplasmic reticulum or rough ER. So this part, if you see the tube-like structures, they are connected to each other. This part is the smooth ER. Why this is called smooth? Because no ribosomes, those dots, are attached. That's why the surface is smooth. That's why we say smooth ER. This part has many ribosomes attached to the surface. So many ribosomes are attached to the surface. That's why the surface is rough, not smooth. That's why it is called the rough endoplasmic reticulum. Okay. the function of smooth ear. If I ask you what's the function of smooth ear, I'll just mention a couple. One is synthesis of lipids, metabolism of carbohydrates, okay, detoxification, what is detoxification? Detoxification is, you know that when we eat food or medicines, a lot of chemicals are entering into our body. Some of those chemicals are very helpful, some are not helpful for our body, some are toxic. When you take the medicine, right, we take the chemicals. And those toxic chemicals must be inactivated, detoxified, and that is done by a smooth endoplasmic reticulum. Okay, so that is an important function. Rough endoplasmic reticulum helps in processing proteins. Okay. You see, the ribosomes are attached to the rough endoplasmic reticulum, and the ribosomes produce protein. Synthesis of protein is done by ribosomes. So if I ask you what's the function of ribosomes, synthesis of proteins. Protein synthesis is the function of ribosomes. Okay. Then after the protein is synthesized, that protein will enter into the endoplasmic reticulum, rough endoplasmic reticulum, and further processing of the protein will be done inside the rough endoplasmic reticulum. What is that? Folding of the protein. Okay, that's just one example. It will fold the proteins, and then after that, after processing of the protein, what will happen? That processed protein from the rough ear will go to the Golgi apparatus. This blue structure is the Golgi apparatus. It's like a number of, tube-like structures, but both ends are expanded like this. Okay, so are packed together. Okay, so that's the Golgi apparatus. You see tube-like structures, but both ends are expanded or round, and if you put them together, that's the Golgi apparatus. In case of endoplasmic reticulum, tube-like structures, but they are interconnected like network. Okay, so that's the difference, structural difference. Now, the protein from the rough ear is sent to the Golgi apparatus, and the protein molecules will go to both ends of the tubes of Golgi apparatus. Protein molecules will accumulate here, and then what will happen? So protein molecules will accumulate here, and then the end parts will get detached and will form vesicle. Okay, so this round sac, membranous sac that contains the protein molecules, that's the vesicle. And inside the cell, you'll find many vesicles. Just you see an example. This is a vesicle that got detached from the Golgi apparatus, and then the vesicle will go to the cell membrane, get attached to the cell membrane, and will rupture to release the protein. to the outside of the cell. Okay? So fascicles contain protein molecules and they rupture to secrete or release the protein outside to the outside of the cell. Okay? So just know those important organelles and their functions. Now we'll talk about the plasma membrane. Plasma membrane is the cell membrane. It is a bilayered structure. That means it has two layers of lipids and proteins formed by lipids and proteins. So it is a double layered structure formed by lipids and proteins. What's the function of the cell membrane? The cell membrane gives the cell a definitive shape. So the shape is given by the cell membrane. And number two, separates extracellular and intercellular fluids. So why that is important? These are the cells and intracellular fluid. That means inter means inside, the fluid inside the cell, and outside of the cell, extracellular fluid. The cell membrane separates those two fluid compartments. That is very important because cell must maintain the internal environment. If there is no cell membrane, then there is no separation between intercellular and extracellular environment. So that is another important function of the cell membrane, giving the shape, and number two, separating two fluid compartments in our body. Okay. There is a theory of definition of cell membrane that is called fluid mosaic model or theory. See here, fluid mosaic theory or model. What is that? Cell membrane is formed by lipid and many protein structures are embedded or present in it. So these are protein structures and whole thing is the lipid. So this is how, if you look from the top, the cell membrane, it will look like this. In the lipid, the protein structures are embedded like this, okay, placed like this. So when you will see from the top, it will look like a beautiful, floor that has many different stones placed in it. Nice floor has many different colored stones or marbles. You sometimes use marbles in the floor, right, to look it beautiful. So it will look like this. The marbles are the protein structures. Now, that's why it is called mosaic. Now, why it is called fluid? These protein structures are not fixed. They always change their shapes. So the shape of the protein always changes. It's not fixed. So when you keep looking, you'll see the proteins are changing like this. Okay, so it's like fluid. Fluid means it's not fixed. Okay, it's changing. So that's why it is called fluid mosaic model of cell membrane. Like the protein is getting small or bigger or changing the shape. So it will look like a beautiful movable floor that's moving. Okay. So we'll see the structure of a cell membrane here. So this is a cell membrane, and this is the lipid part. Okay, this is the lipid part, and these are the protein structures. Now, there are two types of proteins we see in the cell membrane. One type that passes through the cell membrane, throughout the cell membrane, from one side to all the way to the other side. these are called integral proteins. Those proteins pass all the way through the cell membrane from one side to the other side. Those are integral proteins. And you see these proteins are attached to the integral protein, not inserted inside the lipid. These proteins are called peripheral proteins. So those, just I am repeating again, those proteins are deeply inserted in the lipid. Those are integral proteins. Those are attached to the integral proteins from inside or outside. Those are peripheral proteins. Okay, so those two types of proteins we see. Deeply inserted ones are integral. Now, if you see this integral protein and this integral protein, you see the difference is that this one doesn't have any channel to it, but this one has a channel. So the molecules can go in and out through this integral protein. This one will not let the molecules to pass through it. Okay? So when we talk about the functions of the integral protein, we'll see the channel proteins, how they work. Okay. So those are two types of proteins. Now we'll see the lipid part. Lipid part is mainly formed by phospholipids. What type of lipids? Phospholipids form the lipid part mainly. Okay. This is one type of lipid, and this type of lipid mainly forms the cell membrane. Now, if you see the structure of lipid part, it has two layers formed by phospholipid. So that's why we say phospholipid bilayer. Two layers of phospholipid. So I am going to draw the phospholipid molecules or units. So this is one layer formed by phospholipid units. This is another layer. Okay. So one layer, another layer, two layers, phospholipid bilayer. And this is the integral protein. Okay. Then again, we have phospholipid bilayer like this. And this is the peripheral protein. This is integral. This is peripheral. This is phospholipid bilayer. Okay. Now, if we see one phospholipid unit, it is like this. It has a head part and a tail part. Okay. So each phospholipid unit has a head and a tail part. Head part is called polar. It is also called polar end. And tail part is called non-polar. Okay. Polar end loves water. Non-polar end hates water. So which end loves water? Polar. Okay. That's why the polar end is also called hydrophilic. Philia means attraction or love or liking. So hydrophilic is, hydro means water, right? Hydro means water, and philia is loving or liking or yeah. So attraction. So that's the hydrophilic. And non-polar is called hydrophobic. Hydro is water and phobia is fear or scared. So hydrophobic part doesn't like water. It is scared from the water. Okay? So that's the possibility. Okay? So now in the cell membrane, some glucose and other lipid molecules are attached. We'll see those. These green structures are glucose or sugar, okay? And these yellow structures, these are fat or cholesterol. Let me see. Yeah. So fat or cholesterol, and these are glucose or sugar. Now, you see those molecules are attached to the cell membrane. When the glucose is attached to the protein, you know this is integral protein, right? So this is the protein. So when the glucose is attached to the protein, that whole structure is called glycoprotein. Makes sense? Glyco indicates glucose and protein. So glucose and protein together called glycoprotein. Okay? Now you will understand. This is lipid, cholesterol, and this is glucose. When they are attached to each other, we say glycolipid. Glyco is glucose and lipid is the lipid part, cholesterol. Okay? So those structures are also present in the cell membrane. So that's all about cell membrane, structure of the cell membrane that I wanted you to know. Okay? Do you have any question? No? Okay, good. Integral and peripheral, both proteins play some important roles in our body. We'll talk about that soon. Important functions of integral proteins, important functions of peripheral proteins. Just know that those are very important structures. Functions of integral proteins. There are different types of integral proteins. Some help in transportation, some work as receptors, some work as a carrier, and some of them have channels. So they form channels. I have shown you one integral protein before that has channels to it. So some integral proteins form channels. So how integral proteins help in transportation? This is an example. You see, in the cell membrane, these are integral proteins. Both have channels to them. But the difference between this one and this one is this channel always stays open, will not change. It is open all the time. So those molecules are smaller than the size of the channel. They will easily pass through it. Those molecules are bigger than the channel. They will not be able to pass through. But they are always open, right? So smaller than the channel, they will pass through easily. Okay? So that is one. Another type of channel the integral protein has, gated channels. That means they can get closed and open. And to open and close, you need what? You need energy. And that energy comes from ATP. So those are gated channels and they are controlled by ATP, because to open the door, to close the door, you need energy. Just remember what I have mentioned here. So you see that different ways the integral proteins can help in transportation of molecules from one side to the other side. Some integral proteins work as receptors. Very important point. What is that? When you take a medicine, just in that you have taken a medicine, and medicine has chemical molecules, and the medicines should work on the cell of the body, cells of the body. So these are your chemical molecules, medicine molecules, and many integral proteins have binding sites for those chemicals. That means what? If this is the cell membrane, okay, this is the cell membrane, you have integral protein here, you have another integral protein here, okay, and you have another integral protein here. And in many integral proteins, there are binding sites. And just example, this one has a binding site like this. Binding site means it will bind with the chemicals. This one has a binding site like this. This one has a binding site like this. All these three will work as receptors because they have binding sites, so they will bind with the chemicals. Now, you have taken a drug or medicine that has molecules like this. So this one will bind with which receptor protein? This one? Makes sense, right? So because the shape matches. So this will go here and will bind. If it goes there, it will not bind. If it goes here, it will not work. Okay? So the integral proteins have many different types of binding sites and they work as receptors, but specific, very specific receptor functions they will perform. Selectively, they will bind with the drug molecules or chemical molecules. Those will fit nicely with them. Okay, so when that binding will be perfect, this is the perfect binding. If I send this one here, this is not perfect binding. See here, there is a gap. If it goes here, this is not perfect binding. Okay? So when the binding will be perfect, then this integral protein will send a signal to inside the cell. This is the cell membrane, right? So this is outside, this is inside. Okay? So the integral protein will send a signal to inside the cell and intercellular function will be triggered, will start. So just know that how you have seen how the integral proteins work as receptors. And that's why you know that the medicines you take, they work in different parts of the body, right? Not all medicines work on all cells or tissues of the body. You know that, right? You take medicine for your headache, that should work there. If you take medicine for your kidneys, that will work on the kidneys, not on your brain, right? So we know that what kind of receptors are present in which organ or which tissue, so we can make more, could be more specific, right? So that's a nice function of the cell integral protein. Many integral proteins help in stabilizing the cell membrane, making the cell membrane strong. Just know that. How? You see this integral protein provides attachment of many rod-like structures. These are rod-like structures. They get attached to the integral protein, both inside and outside. And it's like we can say that when you go for camping, you put the tent, right? You put the tent and you use the poles to make it strong. Otherwise, it will fall, right? So the poles are like these rod-like structures, both inside and outside of the cell, and they get attached to the integral protein. So integral protein provides support to those rod-like structures to make the cell membrane strong. Just remember that. Some integral proteins work as enzymes. what's the function of an enzyme? The function is converting one type of chemical to another. They help in that conversion. So very simple picture here. They have shown that this integral protein is working one type of enzyme. So it is converting this molecule, chemical molecule to this, and this one is working as a different type of enzyme that is converting this one to this. So just very simple way they have shown that by many integral proteins, the chemicals can be converted from one form to another form. That's why we say they work as enzymes. Okay, so those are important functions of the integral proteins. Functions of peripheral proteins. Intracellular joining, cell-to-cell recognition. Also, peripheral proteins work as enzymes like integral proteins. So, first two functions are quite different than integral proteins. Intercellular joining and cell-to-cell recognition. Those two functions are done by peripheral proteins only, not by the integral proteins. Okay? But the third one is done by both integral and peripheral proteins. So, what is cell-to-cell recognition and intercellular joining? Inter means in between the cells, intercellulars. Joining, those are the proteins. They help in keeping the cells attached to each other. That is very important. You know that when I showed you in last class, epithelial cells. So I showed that. I did not show you a single cell. I showed like this, right? So you see, the cells are attached to each other like this, and your peripheral proteins are here. They help in holding the cells together by attaching the cell members to each other. Okay, so that is intercellular joining. There are different types of intercellular joining. We see 3 different types of intercellular joining. We'll see it soon. Just know that that's the intercellular joining. Hold the cells together. Cell-to-cell recognition. Very interesting, very powerful function of the peripheral protein. What is that? If you receive your body, you know, we do transplantation of tissue, right, from one person to another person. And in most of the cases, what happens? The body rejects. That's the fear. We are scared that when we do transplantation, if the body will accept it or will reject it, right? You all know that, right? When we do the transplantation of tissue, we always think that if the body will accept it or reject it. Why? Because your cells know which tissue is from your own body and which tissue is not. Your body has the ability to detect that. Okay? And that is the cell-to-cell recognition. Your cells can recognize the other cells or other cells. So that is done by the peripheral proteins. Okay? But if I take tissue from your own body and put in another location of your own body, it will be accepted. It will not be rejected because your cells know that which cells are coming from your own body and which are coming from outside. Right? So that is cell-to-cell recognition. That part is done by your peripheral proteins. Okay? And act as an enzyme. I have given example in case of integrals, so I'm not going to talk about that. So just remember those. Now, intercellular junctions. 3 types of intercellular junctions are commonly seen in our body, tight junctions, desmosomes and gap junctions. Okay? Tight junctions are very tight. It will not allow the fluids and most of the molecules from moving between the cells. So tight junctions are very strong. They won't allow the fluid or molecules to pass through it, go from one cell to the next. Okay? It's like heavily sealed. Desmosomes anchor the cells strongly together. Anchored, like desmosomes are like this. From one cell membrane, the desmosome will come like this, and from another one will come like this, and we anchor the cells together like this. Okay, so that's how desmosomes work. You know, in our clothes we use zipper, right? So desmosomes are like this. From two sides, the anchor-like thing will come, and we hold the cells together very strongly like this. Okay. Gap junctions will allow the small molecules to pass from one cell to another cell. Its name is gap. That means there are many holes or pores present in it. So through those holes, the fluid or molecules, small molecules, will move from one cell to the next. Okay, there are many through passages. Okay. So those are three types of commonly seen intercellular junctions. So here they have shown those intercellular junctions. So this is a picture. Again, remember, not all cells have all types of junctions, but they have shown all three in one type of cells. That's rare. That's not seen often. But just for you to see, we see here three different types of junctions they have shown here. Okay, so these are tight junctions. This is desmosome. You see anchor-like things coming out from both sides, and they will go like this. Okay? And this is gap junction. There are many holes, so the molecules will move from one side to another side. Okay? And this is the tight junction. Okay? So if you just magnify it and see, it will look like this. This is desmosome. For desmosome, just remember a couple of things here. I may ask in the test. Of course, if I ask you that zipper-like, attachment will be done by which kind of intercellular junction, desmosome, which one will not let the fluid or molecules to pass through it, that's the tight junction. And if you see the structure of desmosome, this is one cell membrane, this is the next one. So this cell membrane and this, okay, two cell membranes, and these anchor-like structures are getting attached to each other. Those are called linker glycoproteins, also called cadherin. Just remember that. I may ask you. This is my favorite one, so I usually ask that in desmosome, which protein structure get attached to each other to form that attachment. That's the linker glycoproteins or cadherins. And those linker glycoproteins arise from a disc-shaped plate attached to the cell membrane. So that disc-shaped plate, that is called flat. We see that plate, flat disc-shaped plate, that is the plaque, and from there the linker glycoproteins arise. Okay? So remember those two structures. If I ask you, you should be able to answer. Yes?
Did you say disc-shaped?
Disc, yeah, disc is like,
Okay, thank you.
yeah. Okay.
Dr.
Yes. Go ahead.
What did you say about the intermediate filament?
intermediate filament. I didn't say anything like that. Just remember I said two things. One is linker glycoproteins, that is also called cadherins, and that plate, that is called what? Flat. Okay? So those two are the important parts. Okay. Desmosomes very strongly hold the cells together, attach the cells together. And best example is desmosomes are present in your skin cells. So, you know, when your skin gets dry, I believe you have seen that, you can peel the skin, a chunk of skin you can peel like this. When the snakes, they change the skin, you have seen that the big chunk of skin stays together, right? So all cells are strongly attached to each other by desmosome, so that's why we can, they don't get separated, the cells don't get separated, they try to stay together and you get a big chunk of tissue. So desmosomes are heavily present in our skins and also in cardiac muscle, heart muscle. You can write it down. In two locations you will see a lot of desmosomes. One is in the skin. Skin cells are strongly attached to each other by desmosomes. Cardiac muscle fibers, heart muscle fibers. Okay. Transport across the cell membrane. Cell membrane is a semi-permeable membrane. Semi-permeable means partially permeable. What does it mean? If we let some molecules to pass through it, not all molecules will be able to pass through it. That's why we say partial or semi-permeable. It will allow some molecules to pass through it, but will block some molecules too. Okay? Not completely permeable. Two types of transport occur through the cell membrane. One type is called active, another is called passive transport. So active transport and passive transport. Active transport requires energy or ATP. Utilization of ATP is required because it is active and occurs only in living cells, only in living cell membranes, because ATPs are not produced in dead body or dead tissue or dead structures. You know that, right? ATP is produced by metabolism, and metabolism only occurs in living tissue. So that's why for active transport to occur, ATPs are required, and it should occur in the living body. Passive transport does not require any ATP. If you take a filter paper and put in between two different types of solution, the molecules will pass from one side to another side by passive transport. No energy is required for that. So filter paper is a dead structure, right? It's not a living structure. So passive transport can occur through dead membrane or dead. Okay. And in case of passive, why energy is not required? Because the fluid, if this is a membrane, here you have high concentration solution and here you have low concentration solution. Okay? In case of passive transport, the molecules or solutes will always move from high to low concentration. So these are the solutes. Will always move from high to low concentration in passive transport. That means no energy is required towards the concentration gradient. The pressure difference, the pressure is high here, the pressure is low here because concentration is low here. Okay? So pressure of these molecules or solutes is high here. So that will let the molecules move from high to low. The purpose is what here? Remember that, always remember that why the molecules are moving, to bring equilibrium, equal, the concentration equal on both sides. That's the purpose. Okay? So that you need to keep in mind. If you do, then you will understand which way, which one should move in which direction. Okay? So when the molecules or solutes will move from this to this, then it will increase here and will go down here, and eventually equilibrium will come, and then the movement will be stopped. Now, if I want to move the molecules from low to high, then I need energy, because it is against the concentration pressure. So you have to use energy, push these molecules from here to here. That is the active transport. When energy is required, that's the active transport. When it goes towards the concentration gradient, that means from high to low, that doesn't require energy. Okay. The passive transport could be simple diffusion or facilitated diffusion. What's the difference? Another is osmosis. Just remember that those three are passive transports. What are those? Simple diffusion, facilitated diffusion, and osmosis. Okay? So we'll quickly see what are those. Simple diffusion, I have already shown you. The solutes will move from high concentration to low concentration because of the pressure difference, and that movement is simple diffusion. The solutes, I'm repeating again, you can write it down. The solutes will move from high concentration to low concentration. That is called what? Simple diffusion. Right. Facilitated diffusion. For this type of diffusion, just remember a carrier is needed. Carrier molecule is needed, and that carrier will carry the solute from one side to another side. That's why it is called facilitated diffusion, facilitated by a carrier molecule. Like you can move from, if I ask you to move from here to there, you can do two ways. One is you can go by yourself, or you can sit on someone's shoulder, like small kids, right? They don't want to walk. So that's good. They put the kid on their shoulder and they carry, right? So that's the facilitated diffusion or movement helped by a carrier. So you are sitting on that carrier and that is taking you to another side. Okay? Another example, I don't know if you will like it. So you see you are trying to pass or cross a lake or river that has current, and you are going towards the current. So you are being helped by the current. Now, if you know swimming, how to swim, you can, just float your body on water, and since the current is favoring you, it will push you and take you to the other side. Okay? That's one way you can go. Another, you don't need to swim, you just, stay there and the current will take you to the other side. That's the simple diffusion. Because the current, in this case, you see current is this way, from high to low. So that will take the molecules from here to the solid. Another is, if you see a small boat there, you can just sit on that boat and that boat will take you to the other side. Okay? You don't need to use the energy because the current is favoring you, right? So you will just sit on the boat and boat will take you. That's the facilitated diffusion, carrier mediation. Now, if the current is against you, then what you have to do? If you just float your body, can you cross? No, you can't, right? You have to do what? Swim. Or in the boat you have to use engine. That's the energy. When you swim, you have to utilize energy. That's the active process, right? So active process. You are utilizing energy. Osmosis. So, I'm coming back here again. This is a solution, high concentration. This is a low concentration solution. So, in each solution, if I ask you to make a solution, if I ask you to make a sugar solution or glucose solution, you will do what? You will take, which two things you will take? Water and sugar, right? You will take those two things to make the solution. Is it right? Okay. So, just think that this is a sugar solution and this is also a sugar solution, and this one is high concentration, this one is low concentration sugar solution. Now, you tell me, you already know, I have talked about that. So here this is low concentration, this is high concentration. So which one has more sugar molecules, in this one or this one? This one has more sugar, right? Because this is high concentration. That's very simple. So sugar is solute. And what else you have here? Water, right? And water is what? Solvent. So you need to know that when we talk about a solution, you need to know that it has a solute, it has solutes and solvent. Those two things. So in case of sugar solution, the solute is sugar and solvent is water. Okay? So you have sugar and water. Okay, so now this is high concentration, this is low concentration, so more sugar is here. So to bring equilibrium, same concentration, sugar molecules should move from this side to this side. Make sense? Very good. That is simple diffusion. I have already mentioned before. That is simple diffusion. The solutes will move from high concentration to low. Remember, solutes, sugar. Now, also, to bring equilibrium, what can you do? You can move the water. Water is less here, right? Water is more here because this is low concentration. Water is more here, sugar is less. So you can also move water from this side to this side. That will decrease the concentration of this side. Make sense? So when the water molecules will move, the solvent will move from low concentration to the high concentration, that is called osmosis. So both occur at the same time through the cell membrane because our semi-permeable membrane to bring equilibrium. Solutes will move from this side to this side because solute is more here, and solvent, water, will move from this side to this side because water content is more here. Okay? So one is simple diffusion and water movement is the osmosis. The purpose, again, the purpose is bringing equilibrium, same concentration on both sides. So if you know that, you can easily figure out what should be done to bring equilibrium. Both simple diffusion and osmosis should work simultaneously at the same time to bring equilibrium. Okay? This is an experiment that you can do. You see here in an U-shaped test tube, you separate two different concentration solution by a membrane, but that membrane has large holes, larger than the solutes or sugar molecules. Okay? So concentration is higher in this side, lower in this side. So since the hole is larger than the solutes will move from higher to lower. Okay? And will bring equilibrium, same concentration in both sides. That is very simple. Simple diffusion, right? Here, if the membrane has tiny holes, very small, water molecules can pass, but not the sugar molecules. Okay? Now what will happen? Which way the water will move? From low to high or high to low? Low to high, low concentration, because in low concentration you have more water, right? So from low to high. So water will move from this side to this side. But solutes won't be able to move because the holes are smaller than the size of the solute. So you are blocking the simple diffusion, but you are allowing the osmosis, right, in this case. So when osmosis will occur, water molecules will move to this side, and you will see that the level of water is increasing. That means the amount of solution in that side, high concentration side, will increase. And now what has happened, the concentration is equal in both sides. Amount or volume is different, but concentration is equal. Okay? That's the experiment you can do. Okay, now tonicity. Just know that tonicity is very much similar to the concentration. Okay? Relative concentration, that's we say tonicity. Relative concentration. What is that? This is a cell, and this is intercellular fluid. This is extracellular fluid outside of the cell. And if the concentration of the fluid inside the cell is just say 5 milliosmol. Milliosmol is the unit of tonicity. Okay? So it is the unit of tonicity. So inside the cell you have 5 milliosmol concentration in fluid, and outside of the cell you have 10 milliosmol. Now, yes?
Is relative concentration another name for tonicity?
Yes, it is the relative concentration. I am showing what does it mean. Okay? So if I ask you now, which one is hyper, which one is hypo? We are comparing two, right? Relative. So this is hyper, right? Hyper means more, and this is hypo means less. So this solution is hyper compared to this. Make sense? Compared to intercellular fluid, extracellular fluid is hypertonic. Make sense? Now if I ask you, compared to outside, inside fluid is what? Less, right? So hypo. Compared to outside, inside is low. Is it clear? Inside is 5, outside is 10. So this is hypo. Do you agree? This is hypo. Everybody? Okay. Now if I change it to this to 15, is it hypo? What is this? It becomes hyper. Although we did not change the outside, right? So that's why we say relative concentration compared to the other one. If you are more or less. Right? So now if you change this one to 10, that is same, right? Outside and inside, that is called iso. Iso means same. Okay? Hyper is more, hypo is less. Okay? So that's why we say relative. We can, if we change one, another could be changed. Okay. Now if you take different types of solution, hypo, hyper and iso in a container and leave the living cell in it, then how the living cell will react. If you put the cell and inside the cell you have, for example, 5 and outside solution has 10. Okay? Now you already know. You tell me the water or solvent will move in which direction? From low to high or high to low? Water.
Water moves from low to high.
Low to high, right? Remember that? Water will move from low to high. So, from the cell, the water will get out. Make sense? Water will get out. So, the cell will shrink. Okay? Now, if the outside solution has 2, then again water will always move from low to high, right? So water will go from outside to inside. Make sense? So the cell will get bigger and swell up and may even burst or rupture, right? So if you put the cell in hypo, the cell will get bigger and large. If you put it in iso, no change will occur because concentration is same on both sides. No movement is required. Okay? So the cell will remain unchanged. The shape will remain unchanged. So that's how the living cell will react in different solutions. Okay? So if you are a nurse and you are asked to inject saline in patient's body, would you prefer hypo, hyper or iso? Which one you will prefer to inject in patient's body? Patient's blood has different types of cells, right? You will prefer iso because iso will not bother the cells. Hypo or hyper may bother the cells. If you inject large volume, right, that can bother the cells. Okay. So we try to keep the solution isotonic when we inject into the body. Okay.